So
great.
I want to welcome you, and I'm glad that you could give that seminar to us. And as we talked about just now, I think traumatic brain injury, of course, is a kind of and concussions, a topic that has come up a lot lately with sports, but also in environment like veterans and military. And so, but of course, there is a really traumatic brain injury, and there might be a mild brain injury. But what that one is doing, if it's that body, I mean, that might be ignored by most of people. But maybe today we change that. So
Yeah. Well, thank you so much, Doris, for the invitation, and thank you all for attending. Yeah, and I'm thinking about how mild is mild, because I'm gonna show you some stuff that's a little alarming with what we perceive as being a mild brain injury, which is really similar to a concussive-like injury. But really, in the TBI field, people are really starting to question whether mild is a misnomer, because it's still traumatic brain injury. And there's still some pretty chronic changes that occur throughout life. And so it really, it's just, I think we're all moving away from kind of dismissing mild injuries as actually being mild. So the talk of my title is really getting at that sort of these chronic impairments that we see with a mild TBI. But I also really want to take the opportunity to introduce some things to you that you may not know about neurogenesis. And when I was thinking about that, I started thinking about my talk, and I really should have named it like what neurogenesis can do for you, and in turn, like what you can do for neurogenesis, because there's this kind of like just, I think, a very nice relationship where you can definitely do things to modify the good things that neurogenesis is doing for you. But, it's a little bit ironic, because this relationship can sometimes change, and you actually may not want neurogenesis to be as much of an influence for you. Now, I'll talk a little bit about that, and hopefully that will make more sense as I go into my talk more. So just to introduce adult hippocampal neurogenesis, you all are probably aware that your brain continues to generate new neurons throughout your life. This is very different from what I was taught when I went to school. It was very bleak. You're born with a certain number of neurons, and that's it, and that's that. So when I found out that that's absolutely not true, and it's not true for a part of your brain, the hippocampus, that's essential for making new memories, it really blew my mind, and it got me really excited. And I think I've been in love with these little new neurons ever since. So every day, in fact, like you can generate up to like 700 new neurons, and many of these neurons will go on to like mature and integrate, have these beautiful little dendrites. I bring in information about the outside world to the hippocampus and encode them into memory if it's important. And this lovely process begins with these beautiful neural stem cells that live within the hippocampus. When they're activated by granule cells in the hippocampus, they can divide. They'll make a copy of themselves, or they'll make these transient amplifying cells, which then rapidly divide and form these neuroblasts. If these neuroblasts are lucky and don't get eaten up by microglia, they go on to become immature new neurons, where they develop these beautiful branches, these dendrites that again receive information and make all the wonderful things in your brain happen. The really cool thing about neurogenesis, we've been knowing that neurogenesis occurs for decades, since I think the 60s. But people didn't really pay attention to it because it's such a small population of the brain cells in the hippocampus, about 4% on any given day. And people just didn't think that mattered. But when you think about all of the synapses and networking that occurs, it really does matter. And the other reason why it matters is because you actually don't need a lot of cells to encode very specific memories. On the contrary, you need a few to be able to inhibit things and just code very specific information coming in. And so we've learned that these new neurons play really cool roles in different types of memory, not all kinds of memories, but very specific memories. One of them is pattern separation. And the best way I could explain that is just by an experience I think that we've all had. So, you go to someone's house, your friend's house, and you notice that something is different about their living room, right? And pretty soon you realize, oh, they changed the location of a plant, right? Or something random like that. That recognition is due to your immature neurons. So they basically encode very specific information about context to let you know whether something fits with your memory or something has changed, right? And you may not think that this is really important, but imagine that you're somewhere, let's say somewhere you're used to being, right? Um, and let's say that you're depending on very specific cues to tell you kind of where you are, right? You need to know those configurations to not get lost, which is something that often happens as we get older, right? And we know that neurogenesis declines as we get older. And one of the ideas is that this may, in fact, account for some of the reasons why we tend to lose that sort of spatial recognition. Aside from pattern separation, we now know that like they're important for cognitive flexibility, so changing the way that you approach problems. They're important for forgetting. And one of the more recent discoveries is that they're actually important for the formation of long-term memories, and they do that by modulating your sleep patterns. So when you learn something new, your REM to non-REM ratio changes to accommodate that new information. If you ablate these new neurons, you no longer have that change and you no longer have good long-term memory. Okay, we know that they're important for mood regulation. How you're able to buffer your stress responses when something anxiety or fearful happens to you. We know that they're incredibly important for a major class of antidepressants that we use, the serotonin reuptake inhibitors. If you ablate neurogenesis, these SSRIs are no longer effective, so they're critical for the efficacy of antidepressants. And there's pretty good evidence now that they play an important part in injury recovery following various types of brain insults, including TBI. I was telling Doris I was really going to focus on this TBI part for this talk, but thinking about work, like we need memory, right? We need to be able to buffer all the stressful things that happen throughout our work days, and so I think neurogenesis is very applicable to all the things that we need in our occupations, right? Okay, so if telling you that like your brain just on any given day generates about 700 new neurons, if that's not impressive enough, you can actually double or triple this amount by doing things that are good for you. So when you exercise, when you experience novelty, especially like new places, when you learn, all those things make your levels of neurogenesis increase, right? And we know that a lot of those are correlated with better learning and memory. When people are depressed and they take these SSRIs, that increases neurogenesis. By the same token, there are things that you can do to hamper neurogenesis. So if you're not sleeping well, if you're chronically stressed, and paradoxically, brain injuries long-term will cause a depression in constitutive levels of neurogenesis. So just your baseline levels of neurogenesis will start to slowly decline. So I told you that right after TBI, your levels of neurogenesis increase, and that there's this idea in the field that increases is reflective of this endogenous mechanism, repair mechanism that helps your hippocampus recover. And so we began looking at that with a TBI model that's more moderate to severe, a moderate to severe injury. And I'll explain why we were originally interested in that, but we came across some pretty, I think, surprising and interesting, at least to me, it was really surprising coming in new. This was one of the first projects that I worked on with TBI and neurogenesis. And that has to do with this aberrant neurogenesis, which I wasn't expecting at all. And so just to orient you, we do this controlled cortical impact injury. I'm not going to go into great detail other than to say that it delivers an impact with a very small pin directly onto the dura. It sounds pretty invasive because we have to drill a little hole to actually directly hit the brain, but the mice do surprisingly well after this. After a couple of days, like you looking at them through their locomotor, sensory motor behavior, you really can't tell that there's an injury. So we use this transgenic mouse that expresses this green fluorescent protein in new neurons. And what we show is that in contrast or in comparison to sham animals, which animals that don't get injury, on the side of the injury, that's the ipsilateral side, you see this massive increase in the generation of these new neurons, right? And that's quantified over here. So this is a sham animal, and this is a TBI'd animal with this injury. And you could see that the number of these GFP label cells goes up. And that happens on the contralateral side too. So the side that wasn't delivered a direct injury, right? Although it's not as robust. Okay, but when I started looking closer at these cells, I started noticing that a majority of them didn't look like the cells that are born in a non-injured animal. So those cells typically have this one apical dendrite, this one primary dendrite. And as they get closer to the area where they receive signals from the entorhinal cortex, they start branching out. But what I saw in the animals that were injured is that the dendrites didn't shoot straight up. In fact, sometimes they grew laterally. Oftentimes, instead of having just one apical dendrite, they had two. And their dendrites just looked a little bit shorter, even though there were more. And we quantified that just by doing the shoal analysis. And we saw that indeed, the dendrites started growing closer to their cell bodies, and they tended to peter out a little bit earlier. And these are just some skeleton representations of what I was seeing. So again, they're branching out very close to their soma. Oftentimes they have more than one apical dendrite, and they're growing laterally. Okay, and so to me, that's important because, like I mentioned, and you may have picked up on this, these branches need to go out into the areas where they receive inputs, right, about what's going on in the outside world and encode those memories. And if these dendrites aren't going to the places, then it really questions whether they're able to process information correctly. Okay, so the reason why we became interested in this post-traumatic neurogenesis, I said I would mention why we're interested in this. Is everyone able to see my pointer, by the way?
Yes, I can see it.
All right, great. So the reason why we were interested in this is because we wanted a model where we could ask what diazepam exposure does to post-traumatic neurogenesis. And we're interested in diazepam. It's not given in the clinic necessarily, but benzodiazepines are pretty frequently given to people that go into the hospital for TBI. And that's usually just to keep them calm, to keep them sedated while they're intubated so they don't pull things out that shouldn't be pulled out. And so we wanted to know what this exposure does to this post-traumatic neurogenesis, given that GABA receptors mediate increases in neurogenesis and their survival. And so what we did is we did this TBI and a sham, treated animals with vehicle, and in the same paradigm, we gave them diazepam 1 week after TBI, and then ask what it's doing to that response. Is it enhancing it? Is it blunting it? We found out that it blunts it, so it blunts the number of new neurons that are generated after TBI. But the thing that was more interesting to me actually is that not only does it blunt this response, for reasons that I have yet to be able to explain, and this really left my head scratching for a while, is that it rectifies that aberrant morphology in the dendrites that we were seeing, and we quantified this with shoal analysis. In the green, you see these are the guys that were treated with diazepam, and they look very similar to the black line, which are the non-injured animals. And so one of the things that I started then asking is, what does this mean for recovery? What does the aberrant neurogenesis mean? Does it mean that mice actually aren't doing well, and that this increase in neurogenesis, which everyone seems to think is a really great thing, may actually be a bad thing, right? And I should just mention that we patched onto these new neurons just to make sure that they're functional, and as aberrant as they were, they're actually synaptically functional. They're able to receive inputs and put out signals. So that all seemed fine and dandy, and somebody could walk away and say, hey, they're functional, everything's fine, even though they're aberrant, they're functional. And I'd argue that that's the exact problem that we might be having with them, that they're functional, and if they're doing something naughty, they're quite capable of doing it. And so to now sort of ask at a more network level what their contribution is to information processing, we turn to behavior. And so we use the same paradigm where we gave animals a sham or this injury. We gave them the diazepam for 1 week. And then 1 month after injury, we do this reversal water maze, which is really sensitive to changes in neurogenesis. And so in this test, mice are put into this pool of somewhat cold water, which they really dislike. And very soon they learn to find this little hidden platform. It's hidden because the water's made murky with chalk and they can't see it. But they learn its location by using these spatial cues that are just outside of the pool. So you train them to find this platform, and then you do something really naughty and you change the location of the platform. That's the reversal part of it. And so now they have to put these very similar cues, these overlapping cues together in their spatial map, and be able to distinguish between where the platform was located initially and where the platform is now located. And animals that have impaired neurogenesis have a really hard time with this test. They don't typically show impairments in the first part of the test where they're first learning the platform, but they'll show impairments the second platform. And so we trained them. We trained them to locate the first platform. They had no impairments. I'm not showing that, but they were able to learn it just fine as expected. But when we changed the location of the platform, the mice that were injured, that received vehicle, that again had these aberrant new neurons, they had a really hard time doing this test. And when we probed them, that means we took away the platform and we asked, okay, do you remember where the platform location is? Do you spend more time in that area, or are you swimming closer to it than mice that don't have an injury? And so the mice that had an injury, that's CCI'd vehicles, they swim further away from the correct location of the platform, right? And they also made less visits to the location of that platform. But when we gave them diazepam, again, it dampened down that aberrant neurogenesis. Those mice are now performing comparable to sham mice. Okay, so they have a better memory of that platform, and they were able to learn it better as well. That's the guys with the blue line here. Again, aberrant neurogenesis was suppressed here. It was left as is in the red. Okay, so that tells us that this sort of more moderate to severe TBI injury is really resulting in aberrant neurogenesis that actually may not be as good for you as we originally thought. The good news, however, I'd walk away saying that these new neurons can be modified, they can be rectified, and the question is, how to do that. But I'm going to turn now and talk about this mild TBI. And the reason why I want to talk about mild TBI is because it is the most common form of TBI sustained by humans. 80% of TBIs are really concussive-like injuries. And so I told you earlier that as we age, neurogenesis, just baseline levels of neurogenesis go down. And so this really made me think about what's happening to our ability to increase neurogenesis. If we know that TBI is affecting just the baseline levels, does it mean that if you've had a prior TBI, that's going to change how this neurogenic response or the increase in neurogenesis is able to be elicited, right? And so I was interested in that question, not only because of TBI, but also because of all the things I told you either elicit increases in neurogenesis or depend on increases in neurogenesis in order for let's say, antidepressants to be effective. And so to do that, I turned to this mild traumatic brain injury. So here we're delivering a direct hit to the skull using a little piston. And I should just tell you, I'm not showing it here, but I'll tell you that like more than 15 minutes after you give them this injury, if you look at mice, you cannot tell them apart from non-injured mice. They recover incredibly quickly from this injury, and just grossly looking at them, you can't tell them apart. Okay, so in thinking about this, I was like, okay, well, you know, if we know that like neurogenesis reduces or is impaired chronically after TBI, and we know that these new neurons are bad and that they're aberrant, then that might actually be a good thing, right? And why would it be a problem if you're no longer able to elicit more? One huge assumption that I'm making here, and you may have caught this, is me saying that we know that these neurons are aberrant, right? We actually don't know whether they are or they're not. So that's one of the first things that I looked at. And so I used this transgenic mouse that expresses this red protein in new neurons. And I looked at their morphology, and we can't tell them apart from the sham animals. So unlike what I showed you with the more mild, with the more severe injury, milder forms of TBI appear to generate actually pretty normal looking new neurons. And so, yay, that's a great thing, right? And so now we really started to want to get to the question of, does TBI impair the ability to mount a neurogenic response? And so to do that, we either gave animals a sham or a TBI, and 2 whole months later, just making sure that they're fully recovered from that first TBI, we gave them another sham or a TBI. And so animals either had just one sham and then another sham, or one TBI and then another TBI. We injected these mice with a mitotic marker just like 3 days after their second TBI to capture the proliferation of those neural stem cells that support neurogenesis. Okay, and so in animals that got no injury at all, their baseline levels of proliferation are low. These cells don't divide very fast. They're pretty quiescent. They're pretty quiet for the most part until there's some activity. In the mice that got a single TBI, so they got a sham the first time and then a TBI during the second injury. So those animals, we know that a single TBI elicits increases in neurogenesis, so that's our positive control. And we see that this mild TBI does that. It increases the division of these neural stem cells. But in the guys that got 2 TBIs, we no longer see that increase in proliferation. So this is supporting this hypothesis that a prior history of TBI will impair the subsequent response of the neural stem cells to elicit neurogenesis. And just to show you that this does translate into actually deficits in neurogenesis, so it's not just that the neural stem cells are not responding. Somebody could argue, well, maybe the few cells that are generated, maybe more of them go on to become neurons, maybe more of them survive, right? Unfortunately, the answer is no. When we probed for new neurons and let them, we labeled them with this mitotic marker and let them grow and then looked at them. The guys that had 2 TBIs are not showing that there's any more development of these new neurons. So it definitely impairs this neurogenic response. And I should say, that constitutive levels of neurogenesis aren't impaired. And if I go back, and I'll show you the same thing with the neural stem cells. So this is a mouse that got a TBI the first time and then a sham. So it's just showing you the state, basically, of the environment or what's happening just before that second TBI. And when we look there, it's not as if TBI depleted the number of neural stem cells that are available. They're all still there. The pool's not depleted. And it also didn't affect their baseline levels of proliferation. They're still there. So something is happening that's prohibiting the stem cells from engaging in proliferation. So then to ask, why does this matter? We turned to our water maze paradigm. And what we found, so the idea is if eliciting this neurogenic response is important for cognitive recovery, particularly when mice have this normal phenotype in neurogenesis, like their neurons look fine, they're not aberrant. If this is important, then mice without the ability to induce this response should be impaired. And that is exactly what we find. So we did our water maze and we looked at the reversal task, and mice that don't have an injury, right? They spend more time in the correct area where that platform is located. Mice with a single injury are able to do this. Again, those are the mice that elicited a neurogenic response. Mice that had the chronic TBI, so they had TBI for a long time, and then we tested them. They're able to do this test. So it's not just this sort of like development of impairments because of higher inflammation. The mice that had 2 TBIs, again, mice that didn't show a neurogenic response, they're clearly impaired compared to the other groups. So this tells us that losing the ability to engage this response really does make, or it is associated with worse cognitive outcome following a repeated TBI. So the next thing, how am I on time? So the next thing that I was thinking about, I jumped right in and started thinking, why is this happening? Why are the neural stem cells not engaging? They're still there, which is great. They're still able to proliferate, albeit at just baseline levels, but something is happening such that they're just not able to increase their proliferation. And we asked a bunch of questions, whether there was changes in this protein that regulates their activity. It didn't seem to be the case. We asked about preconditioning, if there's like some adaptive response, perhaps the skull got thicker in so that by the time we gave them their injury the second time, maybe it just didn't elicit the same type of response. That didn't seem to be the case. And so I stepped back and I asked a very simple question: does this have something to do with the second TBI and the way that it is being registered by the brain? Or is this something intrinsic about the neural stem cells that's causing them to change their behavior? And so to do that, I activated, or I presented a different challenge. Instead of a second TBI, I allowed the mice to run on running wheels, and they really like doing this. I allowed them to run on running wheels. I told you early on that running robustly increases the proliferation of neural stem cells. And so the question is, if mice had a history of TBI, does exercise still elicit proliferation, or does that TBI actually impair this? And so to do this, we gave animals a sham or a TBI, and then 2 months later, we allowed them to run on running wheels. And we sacked them or euthanized them a few days later to look at the level of proliferation in their brains. And these are just pictures showing the hippocampus again, where proliferation happens in animals that did not have an injury and ran. You could see that there's proliferation happening, but that's really reduced in animals that ran but had a prior TBI, and that's quantified here. So again, animals that are not injured and run show higher levels of proliferation compared to when they don't run. But animals that are injured despite running don't show higher proliferations than the animals that are injured but did not run. And it's not because perhaps TBI'd animals ran less. When we look at comparable levels of running, they're still proliferating less. So this also tells us that this impairment is not likely due because of adaptive changes, again, like the thicker skull or maybe differences in inflammation, because unlike a second TBI, running doesn't induce proliferation through inflammation. So one kind of cool clue that this gave us is that both TBI and exercise should be eliciting vasodilation, so they should be increasing blood flow. And I'll get into exactly why that's really important for this. But I went back and looked at just kind of the vascular health of animals after their TBI. One kind of very silly assumption that I made early on, because animals recover incredibly quickly and you just cannot tell them apart from sham animals. My mind was set on this being a mild TBI. And so I never really bothered to closely look at their brains to see if they had obvious defects. And one of the most common and frequent impairments after TBI, which is this impairment in the blood-brain barrier. So things start leaking out of blood vessels onto the brain that shouldn't be there. And that elicits a whole host of problems. So when we looked at the blood-brain barrier, we injected this dye, Evans blue, and it usually doesn't cross into the brain from the blood unless there's leakage. And so we looked at our blood vessels early on after this mild TBI, and we do see evidence that they're leaking. It wasn't all that impressive. And so here I go again with my assumptions, thinking, well, later on, this should heal and not look bad. If anything, that leakage is actually looking more pronounced. We have yet to quantify this, but we are working on that. But just looking at these, it looks like this leakage is actually getting worse as time proceeds. We then looked at this marker, MFG8, which is a pretty common marker that people in the vascular field use to look at vascular remodeling. It's a pretty good indicator that there's some injury going on in the blood vessels. And so we looked at the blood vessels stained here with this marker CD31 that labels endothelial cells around the blood vessels. And we see pretty prominent and consistent, persistent elevation in levels of this MFG8 in injured mice compared to sham mice, and that's quantified here. So we see greater intensity, and we see that more of the blood vessels have elevated levels of MFG8. So I mentioned that I would say why this is really critical, why impairments in the vasculature. I mean, that's kind of a no-brainer, no pun intended, but we know that the vasculature is important for brain health, but it's particularly important for the activity of the neural stem cells. And that's because these neural stem cells wrap themselves pretty aggressively and tightly around capillaries to receive the oxygen and nutrients and the trophic factors that prompt them to divide and sustain this high metabolic need for oxygen and nutrients. And so if you're messing up the delivery of these factors, I think it could be a pretty good explanation as to why these cells they're okay. They're able to sustain baseline levels of proliferation, but they just don't have what they need in order to enhance that proliferation. And so as proof of principle, if this is a vasculature problem, then we reasoned if we induce a vascular injury with angiotensin II, which is pretty involved in vasculature impairments, if we induce vascular impairments, do we see that we now are able to mimic the impairments after this second TBI? Do we cause the first TBI, which usually is perfectly fine and able to elicit this response, but if we induce this vascular injury, is that first TBI going to be able to still respond? So we did that by giving animals vehicle or angiotensin II, and I should say this is a pretty low level, or it's called a slow pressor dose of angiotensin II. It doesn't actually interfere with like mean arterial pressure, but what it does is it makes vessels not reactive. It doesn't allow them to dilate very well. And that's exactly the type of impairment that we think we have. So we induced this impairment, and then we gave a single TBI, and we asked about the proliferation. And animals that weren't given the angiotensin II, so where we didn't induce this vascular impairment, we see that they have a pretty good response to the TBI. They proliferate just fine. But in the animals where we induce this impairment in vasodilation, they show deficits in their ability to respond to the TBI. And this is just a picture of this angiotensin I receptor in animals that have had a TBI, and we looked at them 2 months later. So they show elevated levels of this angiotensin II. Okay, so we really started going after this idea that the vasculature is impaired. And so to look more closely at it, we did this protocol where we wanted to isolate the endothelial cells around the blood vessels, and we're able to do that quite nicely. We wanted to run Western blots and later on do like some RNA sequencing to see what's changing about them. What clues can that give us to try to figure out how they're being impaired? And so we did this protocol to isolate them. It's basically taking the brain, homogenizing it, and running a density gradient with dextran to separate the blood vessels from the rest of the brain. And like I said, we're able to do that just fine in the sham animals. We can enrich our blood vessels. So if we run a Western blot to look at protein for that is expressed by these endothelial cells, you can see that that's enriched in our blood vessel fraction. So we have more of those cells in the blood vessel fraction compared to the homogenate. And that is great. But when we do the same thing in our TBI'd animals, we look at the homogenate here, and we look at the blood vessel fraction here, we don't see that same degree of enrichment. And it's not because there's less endothelial cells. That was my very first thought, that there's maybe like the endothelial cells are dying and that's why we don't get an enrichment. There's actually no difference. We've done this now a good number of times. There's no difference in the starting quantity of endothelial cells. So homogenates have the same amount of CD31 in the homogenates. But when we isolate them, they're not there. So this tells us that we're losing these endothelial cells in the fractionation process. And so this really hints at a problem in their ability to remain attached. It's not that we're losing them, it's just that they easily detach from the blood vessels. And so this can be a really big problem for dilation, because the endothelial cells are required to anchor. They need to anchor appropriately to the basement membrane in order to either constrict or dilate the blood vessels. And they're able to do this through this integrin receptors. And so to just very quickly and preliminarily ask whether this is a problem, we use this drug, semaglutide, that enhances the activity of integrin receptors. And we just went straight forward. We asked whether enhancing the activity of these receptors improves proliferation. And so previously, I'm going back just as a refresher, going back to this data where I showed you that animals with 2 TBIs or repeated TBIs are no longer able to increase their levels of proliferation. But when we give them semaglutide prior to the second TBI, so we give them semaglutide 1 week after the first TBI and prior to the second TBI, they are now able to elicit increases in their proliferation that's comparable to mice that have just 1 TBI. So it restores their proliferation. I should be the first to say, although semaglutide is incredibly selective for these integrin receptors, these integrins are also expressed on other cells, not just endothelial cells. And so we are now working on a grant to try to figure out whose integrin receptors are disrupted, and really if the cerebral vasculature can explain why we lose this response. Okay, so I've kind of told you quite a bit. I'm just going to summarize. So early on, I told you that a prior TBI impairs the neurogenic response to a repeated TBI, and that this then hampers or impairs strategy flexibility, the type of learning that's needed for these mice in reversal water maze learning. I showed you data that indicates that this isn't restricted to just a subsequent TBI, that it also impairs the ability of exercise to induce neurogenesis. And so one of the things that I'm very curious about is, how much of this actually extends to other things that require neurogenesis or elicit neurogenesis. I've told you about injury and about exercise. I've told you about antidepressants, but neurological injuries may also depend on neurogenesis to provide some resistance against the pathology that they elicit. So very early on in Alzheimer's, for instance, it's very interesting that neurogenesis actually increases, and there's this idea that this challenge of pathology associated with Alzheimer's is actually eliciting this response, right, as a way to try to continue to function normally. And so one of the things that I'm really curious about is, if you've had a prior TBI, are you going to lose that ability to provide resistance against Alzheimer's? And is this why people with a history of TBI have earlier onsets of Alzheimer's pathology? And does that actually provide some clue about the link between TBI and Alzheimer's? I've shown you some evidence that this may actually be a vasculature problem. So we see elevated levels of MFG8, which again is a good indicator of vascular remodeling. We showed that blocking MFG8 activity, so MFG8 actually binds to these integrin receptors at high levels. It inhibits these receptors. So blocking MFG8 activity actually rescues it. Of course, this is an assumption. I haven't shown you that we've blocked it, but we have shown that it does increase these levels of proliferation. I showed you this proof of principle experiment where if we induce impairments in the ability of vessels to dilate, it impairs the neurogenic response. And I've shown you that we are seeing some evidence of endothelial cell vulnerability. Okay, so this has been a little bit of a roller coaster, right? Where I show you something bad, that moderate severe TBI leads to this aberrant neurogenesis, which is kind of boo, right? But I've also shown you that there is a way for this to be rectified, which is great, which gives us some sort of promise, right, to try to intervene in this process and actually make better use of these new neurons. If we can rectify them, will that increase become useful? I've shown you that neurons born after a more mild TBI, which is, again, the most frequent type of TBI that we experience, those new neurons appear to be normal, which is, yay, great. But that even a mild TBI can impair the ability to elicit their increases later on, which is bad. And also, I've shown you that impairments in the neurogenic response may actually contribute to worse outcomes following repeated TBI. But the good news is that we do have some pretty good indications that this may be a cerebral vascular dysfunction. And out of all the things, all the sort of mechanisms that I've looked at, one, this really appears to be the most promising one, but I'm also, if this happens to be one of the primary ways that this is being impaired, I'm excited that it's this one because we have ways to improve cerebrovascular dysfunction. And so can we take, if this does happen to be one of the primary mechanisms, can we take advantage of currently FDA approved drugs to improve outcome and restore this important process? And so one of the takeaways that I want to leave you with is really sort of captured in this one picture. Make sure that you exercise as much as you can regularly. Go to novel places. You want to make sure you sleep well, hopefully better than this little guy. And please just protect your head when you engage in risky activities. With that, I absolutely have to thank a lot of people over at OHSU who helped start these studies and all the support that I get at my new institution, Legacy, for helping me continue these studies. OPAR for funding the studies with diazepam. And with that, I thank you. And I'm happy to take any questions or chat about any of the things I showed you.
Okay, so thanks you at first. And if someone has questions, I think just open your microphone. Can you see the chat?
Yeah, let's see. Thanks for the clarity of the presentation. Thank you. It disappeared. Hang on. Let me see if I can chat.
Are there transitory changes in general blood-brain barrier permeability that may not occur in a normal aging process? Can you see it again? Or
yeah, in the BBB that may not occur in the normal aging process. Yeah. I don't know the degree of leakage, to be honest. I don't know if you know, Doris, just how much leakage there is, but this is pretty profound. I don't know.
Normal aging, I don't think there's that much. I mean, that's clearly in like Alzheimer's disease, and you have like some blood-brain, but
Yeah, and that question kind of prompts me to I'll just very briefly talk about one of the mechanisms that we thought may be happening. This obviously, when we first saw it, looked like accelerated aging to us. I told you that as we get older and neurogenesis declines. Thankfully, in humans, it doesn't happen as rapidly as in mice. So that's good. Keep doing fun, novel things. So what we thought it may be just like a accelerated aging, and one of the assumptions again that I had with that is that older animals did not have a neurogenic response. I know that neurogenesis in animals like goes down really, really fast, especially between 2 to 4 months of age. By 9 months of age, they have very little neurogenesis. So we took some mice that were 9 months old, and we asked, are they capable of eliciting a neurogenic response? Good news for us is that they do it robustly. So even if our levels of neurogenesis go down, we're still able, or at least mice are still able to respond to TBI. And so it doesn't seem to be an issue with senescence. And so how much this actually parallels with aging is a good question, but it's not one that I feel right now is really worth pursuing all that much.
Anyone else? So I actually might ask a good question. So in the more severe TBI, I mean, you clearly also have damage of vessels. Nevertheless, they seem to crazily proliferate. So is that kind of maybe because they are so much damaged? Is that actually
Yeah, so
you know?
So if I understand you correctly, you're saying that in the more severe TBI, you get a pretty profound increase in proliferation neurogenesis, right? But that's to one TBI. So just with one TBI, regardless of whether it's a mild injury or more severe injury, you're going to get a response. The question, I guess, should be like, if I were to give another TBI, right, a more severe TBI, would it elicit the same response? So we did that, actually. We gave a mild TBI initially, and then we gave a more severe TBI, that CCI model, and it's still impaired, right? And we did that for that very question, right? Whether a more severe TBI would elicit it. But also to ask if it has something to do with the skull getting thicker. And so with the more severe TBI, you do a craniotomy, so you basically circumvent the whole skull question.
So and then the epithelial cells on the blood vessel, I mean, it's also happening in the contralateral side, right? So there must be some talk throughout the entire brain, basically.
Well, yeah, it's that, but again, like I've learned so many lessons about assumptions with this project. The contralateral isn't immune from like the mechanical injury itself, so it's getting injured, right? It really is a pulse that kind of ricochets, and I think that is probably one of the reasons why there's so much heterogeneity in the TBI. Because, no one injury is going to elicit the exact same pathway.
No. Okay, someone else? Okay, if there are no more questions, then thank you again for doing this.
Thank you so much. And thanks for the good questions. Appreciate it.
Okay, bye.
Thank you very much.
Thank you very much.
Thank you.
Bye.